Hello everyone, welcome to my channel. In this video, we are going to discuss about the suicidal bag of organelles or the suicidal bag of cell, which is nothing but the lysosome. Lysosome are the membrane bound organelles that are found in the cytoplasm of both plant and animal cell. So, in this video, we are going to discuss about details on suicidal bag or lysosome of cell. Before going to the discussion, if you are not subscribed my channel yet, please subscribe and stay tuned with. So, lysosome was discovered by Belgian cytologist Christian de Duve in 1955, and the word lysosome is derived from two Greek words. First is lysis, which means breakdown or destruction. Another word is soma. Soma means body. So, in combine, lysosome is the destructive body, and it helps in intercellular breakdown of substances. They are more commonly found in animal cell, while only in some plant groups like slimodes, lower group of fungi, etc. This is the structure of a lysosome. Actually, lysosome doesn't have any distinct shape or structure. That's why lysosome, the nature of structure of a lysosome is pleomorphic, means doesn't have a distinct shape or structure, but mostly globular or granular in appearance, which we already seen in our first slide. So it is about 0.2 to 0.5 micrometer in size and surrounded by a single protein membrane in composition. Here is the lipid bilayer membrane, where outer part is the globular protein head. This is the two-layer membrane and outer part is globular protein head. So, outer layer is lipoproteinous membrane. So, outer wall is globular protein. The membrane consisting highly glycosylated lysosomal associated membrane protein. This protein of outer layer of lipid bilayer membrane is called LAMP. That means lysosomal associated membrane protein and lysosomal integral membrane protein. Outer layer, inner layer or protein layer is a circular outline. These are the two types of protein. First is LAMP and next second is LIMP. So, lamps and limps form a coat of the inner surface of the membrane, and they protect membrane from attack by numerous hydrolytic enzymes retained inside. These are the hydrolase means hydrolytic enzymes are here. So, this layer is a protective layer protective layer against hydrolytic enzyme. The lysosomal membrane has a hydrogen proton pump. Here is the transporter, and here is the ATPase means hydrogen pump, which is responsible for maintaining pH conditions of the enzyme. The acidic medium maintained by proton pump that pumps H plus inside the lumen. Here are the H plus ion, and these are responsible for maintaining the pH. Here is the proton pump. Inside the membrane, the organelle consists of an enzyme in the crystalline form. These are the crystal form, looks like a pellet. So hydrolactic enzyme or hydrolase available inside the lysosome in the form of a crystal. Next is the types of lysosome. Lysosome are of two types, that is primary lysosome and secondary lysosome. Primary lysosomes are sac-like structure enclosing enzyme. Here is the storage granule or a primary lysosome. This one is the primary lysosome, and it is synthesized by rough endoplasmic reticulum. Here is the rough endoplasmic reticulum which synthesizes the primary lysosome, simply called storage granule. Next is the secondary lysosome, formed by fusion of a primary lysosome with phagosome. When primary lysosome are fused with phagosome, it leads to formation of secondary lysosome. So here is the secondary lysosome. Here is the phagosome, and when here is the primary lysosome, and this portion is the primary lysosome, and here is the phagosome. When this phagosome fuse with the primary lysosome, it becomes secondary lysosome. Contain engulfed material, so it contain engulfed material plus enzyme here inside the secondary lysosome, which is formed after fusion with phagosome, carry or engulf some debris or materials plus enzymes. Materials are progressively digested. Those debris or unnecessary or foreign materials are engulfed by these are later digested inside the lysosome. Okay. So this is the primary and secondary lysosome. Secondary lysosome, and here is the primary lysosome. Next part is the function of ribosome. Function of, here is the spelling mistake, let me clear. The main function of lysosome is digestion and removal of waste, means debris of cell or foreign particle of the cell, especially the debris of cell. Somehow it also digests the unwanted particle inside the cell. Cellular debris or foreign particle are pulled into the cell through the process of endocytosis. By the process of endocytosis, it pulls the debris or unnecessary material inside to the lysosome. So here is endocytosis, means those debris or foreign particle are come inside the body are digested by lysosome. The process of endocytosis happens when cell membrane fall into itself. It form a invagination. Here is the loop formation called a invagination, creating a vacuolar or pouch around the external content and then bring those content into the cell. This is the pouch-like structure. So here is the endocytosis. It forms the invagination in external wall, a pouch-like structure, and the debris are entered and digested by lysosome, then removed by residual bodies through exocytosis. Those residues or debris are not removed by the process of exocytosis or comes to the autophagic vacuole. Here is the autophagic vacuole. And these are digested inside this autophagic vacuole. The process of autophagy involves in disassembly or degradation of a cellular component through a natural regulated mechanism. Here is the natural mechanism who digests this unwanted debris of the cell by the process of autophagocytosis. So, I hope you are clear about endocytosis, exocytosis and autophagic vacuole or autophagocytosis. Also, lysosome remove junk that may accumulate in the cell, helping to prevent a disease. So, here we finish with the structure, types and function of lysosome. I hope you people are clear with. If you have any query, please comment in the comment box.